Good afternoon, everyone. Welcome to our inhalation webinar. My name is Emma, and on behalf of Oxford Global, I would like to thank you all for joining us today. Today, we will have Josué Sznitman, Associate Professor at Technion, Israel Institute of Technology, who will be presenting part 2 of point targeting in the lungs using inhaled magnetic aerosols. After the presentation, we will have a 10-minute Q&A session. Please submit your questions through the chat option on the bottom of your menu display. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. Without any more delays, I would like to get started with Josué's presentation. Josué, over to you.
Thank you very much for the opportunity to give kind of a follow-up of some progress of the work we had an opportunity to present both through webinar and last year at in London in May. So without further ado, I'm going to take you by the hand and give you a little bit a recap of what this story is all about. Just a word of acknowledgement, if I may, with regards to who's actually contributed mostly. So the work you're going to see is very much the baby of my former PhD student who's actually still working on this project. So to the left, my former student Jan, and the lead engineer here, Semyon Dorfman, who really have been carrying this through. Just a quick word that this work has been supported both by the European Research Council, the Israel Innovation Authority, and is actually part of a European network funded by the Cost Actions called Siminhale, for those of you who may be familiar with. So very briefly, so why on earth are we interested in the concept of point targeting in the lungs? There are a number of diseases that are initially localized, and one can think of a few. I've shown here a few examples. It could be a fungal infection. It could be a bacterial infection, such as pneumonia, where the onset would be in a specific lobe. And of course, we have in mind cancer tumors, which you can see on one of the CTs on the bottom here. So the onset, at the very least, is very localized, and this would not call for generalized drug delivery, but rather targeting, at the very least, a region. So if we go back to the books and think of aerosol targeting in general, if you think of the state of the art, so I'll draw your attention to the left-hand side, which is kind of these classic plots of total deposition on the y-axis versus the diameter or the size of particles on the x-axis. Please note that these are log scale on the x-axis. You see here kind of a staple of drug delivery, which is this kind of a valley-shaped deposition curve. And again, this is total deposition. And the thing that you should keep your eyes focused on, other than the fact that these three curves that have the same trend are picturing different inhalation rates, is that when you think of therapeutic aerosols, kind of the off-the-shelf therapy, and by far, by large, drugs are of the size, basically give or take, in the 1 to 10 micron. And what that means is that most of this stuff is going to be deposited somewhere inside the lungs. And that's why you very rapidly see very high deposition rates. Now, that needs to be, there's sort of two caveats to this kind of generalized drug delivery approach. One, and you can see it on the top right-hand image, which is gamma scintigraphy. So they're basically radio-labeled aerosols that are showing you a snapshot in time of where deposition is. And they're showing you that you indiscriminately deposit aerosols a bit everywhere. The drawback to that is that when you look at kind of a disease scenario, and I'm taking somewhat a more drastic example with cystic fibrosis, is that actually this indiscriminate targeting strategy works somewhat well when you're healthy, but if you have obstructions, if you have some kind of regions that are not ventilated, then actually you're depositing drugs exactly where you don't need them, and this is what the picture on the bottom is showing you. So you flood the lungs, but on the other hand, you don't really get where you necessarily need them to be. So let me kind of put a framework around what we're proposing here, and that's a targeting strategy. So keep in the back of your mind that to this very date, there are still no methods for a targeted delivery. And when I say targeted, I mean very much lobar, if not point, okay? So I'm referring back to the concept of, for instance, a malignant lung nodule or a cancerous tumor, which really starts off as a mass. And the recipe here is to achieve this local targeting. We're going to need, one, a concept of a smart inhaler, which I will elaborate. Two, the idea of leveraging magnetic particles. In particular, I'm referring here to spions, which are super paramagnetic iron oxide nanoparticles. In fact, they are FDA approved, so it's not something new. And three, obviously, that we're going to need some sort of a magnetic field. In this case, we're talking about an external field. So I will elaborate a bit on this. Let it be clear that by no means our work is a standalone. And what I mean here is that people have been working in this field, and the concept of magnetic targeting is in itself not new. I will highlight one example that's perhaps kind of the hallmark of this of the attempt in vivo, and the idea was basically to have an external magnet located on the external thorax of rodents on one side, so one lung, and comparing it to results on the other lung. And the idea here was rodents were inhaling nebulized suspensions of droplets in the range of something like 3 to 4 microns that were loaded with these spions. And the increase that they got, and this is what you're seeing through this histological cut on the bottom, magnet versus no magnet, what you're seeing in black are actually the spions, aggregates of spions inside alveoli, was that they could get something like a 2-fold increase between no magnet side to magnet side, which is good, but still puzzles us with whether or not all of this really is worthwhile when the relative increase is quite feeble, one must argue. And not just that, but the truth is there were never any further studies that were taken on into larger animals, and I'm not even mentioning here humans. But on the other hand, this was kind of a first really kind of splash of where this was taking shape. Now, there are basically 3 challenges that have prevented or jeopardized the translation of a successful targeting strategy. And we believe we've identified them, and they're all linked to very physical mechanisms. And the first one is, as long as you inhale aerosols, you can't stop the drag forces. And what we mean by that is that the reason why aerosols are flowing is because the drag force of the wind, of the airflow that you're inhaling, is pulling these particles into your lungs. So as long as you breathe, they will be flowing. And there is no magnet, no reasonable magnet, that will be able to basically stir away the particles from the flow. It's just too strong, and the only way, and I'm sorry, let me draw your attention to the right-hand side. What you're seeing there is a study where they basically wanted to do tracheal delivery, targeted delivery of aerosols with an external magnet, which you're just seeing here in a schematic fashion, and in black are the deposited particles. And this snapshot is showing you that as long as you're breathing, the bulk of the aerosol bolus will always just fly by. So there's a very simple solution to basically alleviate this problem and just bring it to zero, which is hold your breath. If you hold your breath, then basically you are switching off the drag forces induced by the airflow. So that's the first challenge, and we'll talk about it a bit later. The second challenge is a concept, which is if you have a magnet that's positioned outside of your lungs, surrounding your chest cavity, then the magnetic forces will, of course, always be stronger nearer to the magnet. And what I mean by that is that if you're inhaling continuously an aerosol bolus, then if you pass beyond the point that you want to target in the lungs, then you will be witnessing deposition in regions that, again, you don't necessarily want there to be any deposition. So what we're proposing here, and I'll play this short video, you see a short aerosol bolus that's kind of chopped, and reaching the depth of this tumor that's highlighted in the, that's highlighted, I will play that again, pardon me, that's highlighted in red as if it were a little ball, kind of a fungal ball there. And the idea is that you want to monitor the depth to which the aerosol will reach. So you're tracking the volume of air that's behind. If you don't really go beyond that depth that you want to target, then anything standing between the depth and the magnet will not see any deposition, because simply said, you will not have brought any particles there. So that's the second challenge. So one, drag forces will always win over any magnetic force. Two, you don't want to have deposition near the magnet, and so what your job is to bring the bolus only up until the point of deposition, which is here comprised by the red point. And three, very importantly, is that unlike traditional aerosol therapy, you want to minimize particle deposition. Now, what do I mean by that? If you think again, and I'm bringing back this classic plot, we looked at where the therapeutic aerosols are. Most of the time, you get 50 to up to 100% deposition, depending on the aerosol sizes. Of course, it's indiscriminate. It doesn't mean I'm giving you in this picture where in the lungs they're deposited, but the point is they stay in, and you do not want that. In particular, think of chemotherapy. You only want deposition where the particles are needed, in this case, the lung tumor. So actually, what we're proposing here is that you want to pick the size that is the worst for traditional drug delivery, which will be the best for targeting, which means anything that will not be deposited at the target point will just be exhaled. And what you're seeing here is you want to pick the valley. You basically want to pick particles that are somewhere in the range of half micron, because those do not diffuse sufficiently, nor are they heavy enough to fall. So there's no time to basically deposit all over the lungs. This is the kind of traditional smoke, the particle size of smoke, which is, say, for instance, what you would see in smokers. When you inhale, the bulk of the cloud is exhaled, and that's exactly what you're witnessing. So let us recap this through this short cartoon. We are talking about point targeting. What you're seeing here is now the inhalation phase of the bolus. It's a short bolus, right? We have a controlled depth. It's going to stop there, controlled depth, breath hold, and now will be the time to set on the magnet, which is just a schematic form here. That's not the embodiment of the real magnet. It will be turned on. These are spion-loaded aerosols, and so you will get a magnetic field that is focused in the region of the magnet, and then you turn it off and you exhale. And again, you exhale everything else because, as I've told you, you've chosen a particle size range that will not deposit by diffusion or gravitation during that time. All right. So, right, seeing is believing. This is not in vivo yet. We are showing you hot off the bench, basically our in vitro proof of concept, which supports the engineering solution. All right, so I'm not going to dwell too much into a course of forces and mechanics, but there is always gravity, drag, which happens as long as you breathe, and of course the magnetic force. So if you hold your breath, suddenly drag from the wind, from your airflow, is going to be minimized. And with that, what you end up with is basically a kind of a battle, a relationship between gravitational forces and magnetic forces. And your job is to maximize this ratio of how strong the magnetic force will be against the natural gravity of your particles, even if they might be weak. Okay, Now, what do I mean by the magnetic force? And we don't need to have a whole crash course on magnetism, but the thing I want the audience to keep in mind is that the magnetic force that these spions will experience, and please bear in mind that on their own, these spions experience no magnetic force. They're iron oxide, I mean, they're passive particles unless they're in a magnetic field. So this magnetic field now is the real key to the story. And for those of you who are not so familiar with equations, and that's perfectly fine, the point I want to draw is there are two elements that guide the strength of this magnetic force. One, the magnetization of your particles, which is pictured through the graph on your right-hand side. So you want to have metals, so to speak, who are kind of super magnetized. This is where you see this kind of hysteresis curve that saturates. And the other thing is you want a magnetic field. But what's important is not just the amplitude of this magnetic field, but the gradient of this magnetic field. So you want regions that are changing in the magnetic field, and this will strengthen, enhance the magnetic force. So you need to think that you don't want a constant field, you want changes. You want a place where you go from little to a lot. All right. A word about the in vitro experiments, and this is basically a schematic of them. The black box that has this open window on the left-hand side is our quote-unquote smart inhaler. The idea, so this is a custom designed inhaler. Its job is to create this short aerosol pulse. The embodiment of the delivery is thought as a first proof of concept of a chemotherapy that would be delivered to an intubated patient. This is not the only way this technology can be implemented, in that you could have patients complying with kind of a standard inhaler, but for the proof of concept, it must be said, easier to think of a delivery system where we control the flow rate through a ventilation machine, which is what you're seeing on the top right-hand part. You have a mechanical ventilator that's talking to the smart inhaler, which is delivering the volume behind the aerosol bolus. On the bottom part, what you're seeing is basically an endotracheal tube that is fitted into the in vitro patient with an upper airway model and, of course, the magnet on the left-hand side, shown schematically. Okay, so where does this bring us? It brings us, next, to the in vitro model. Just a very, very short word about our in vitro model. This is coming from classic morphometry, classic studies. The idea was to make a kind of a average human airway model, about 7 generations. Here it's intubated, so that's why it looks cropped on the top-hand side. And the key is that this aerosol bolus needs to be controlled. And for a controlled, tight aerosol bolus, you need to minimize turbulence. And for that, you need to have relatively slow, steady ventilation so that your flows are in fact laminar. And what we've looked at here is in a very simple embodiment with a ventilation distribution that's homogeneous. And again, we acknowledge the fact that that's not necessarily the physiological case, but we're showing the proof of principle of the targeting part. All right. The other thing is, I'm coming back to the fact, what about these particles? So what we did is we loaded a nebulized suspension of water with some salt and about 3% spions. And the basic spion is about 10 nanometer large. We've used an assembly of permanent magnets. And again, just a word about the flow and aerosol pulse. These are very slow flow rates, okay, in this mechanically ventilated patient. And again, the slow flow rates are only conducted during the administration procedure. So it's not, you don't endlessly ventilate the patient at such flow rates. It's just during the administration of the bolus. The pulse duration, which sets basically the width of the aerosol, how big that little bolus will be, is set to a very short time of 250 or so milliseconds. if you look on the right-hand side, you will see a particle size distribution according to mass fraction for the aerosols that are coming out of our smart inhaler. And what you will see is that actually they really park around the half micron size. 90% of our particles are under 1 micron, and so that fits that challenge number 3. If you recall, what we want are particles that fit in the valley of deposition. In other words, they minimize deposition. Okay. So now a little bit of kind of seeing is believing. So the first level challenge is the creation of a bolus that's tight, that will not disperse, that has little sedimentation, that has a controlled length, and that can basically reach a specific position. I'm going to play this movie. What you are seeing here is, what you are about to see, excuse me, is a video of a glass tube that has the dimensions, in particular the diameter of an average adult trachea. The green light is a laser sheet, a green laser sheet that is passing through the tube. That's why you see reflections on the edges, which will help visualize in real time. So the movie I'm about to play is happening in real time as we speak, of the bolus passing by. Okay, so no magnet yet. We're just going to look at this aerosol bolus. Oops, I'm sorry. I need to play this video. So there it is. We brought it exactly to that position. There's a marker. And we're not playing loops here. We're playing a next bolus that's going to be shot. And you can hear. I'm sorry if the voices are overlapped. What you're seeing, every click is that those valves opening and closing that are chopping the bolus so that it's tight. And it looks like a rocket, like a bullet-shaped bolus, and it's not dispersed. Okay, so what it means is that you can control the flow and the bolus shape. Those are the aerosols we discussed. Okay, so that's permanent magnets. On the left-hand side is the ventilator side, and I've just showed that we're going to inhale the bolus, it's going to deposit, and we're going to exhale it, because remember, if you think of the cartoon we saw, we will want to inhale, and any superfluous aerosols will be exhaled so that we don't have any other deposition. So here we go. So it's depositing, and you see that things that are not in the vicinity of the magnet are just exhaled out. So once again, you inhale, the bolus comes to the region, deposits, and exhalation phase. Okay. So what are you seeing now? We wanted to really highlight that this is precise targeting. We basically deposit particles only in the vicinity of this external magnet, and the precision is within less than a millimeter. So the bolus is depositing exactly where we want it to go. What you're seeing on the right-hand side in this rendering of the tree, what we did is we did fluorescent microscopy to count actually all the particles deposited. We added a suspension of fluorescent particles to the nebulized suspension and minutely went through counting all the deposited particles by inserting transparent sheets that were rolled up into the model and pulled out and quantified under microscopy. And so you see the density of the number of particles, their nearest neighbors, of where the bolus has deposited. So if you think of that slide I showed you about if you breathe and you're trying to get tracheal deposition, as long as you breathe, you will just have most of the aerosols passing by. Well, we're showing here that actually, if you want to do tracheal deposition, you can nail it. You can really nail it with the combo of the short bolus, holding your breath, and the right particle size range. Okay, so the point I also want to draw is coming back to this challenge 1 of the drag forces. This is the same concept. You have the magnet here, but you don't implement a breath hold. And as you see, the bolus is barely deflected. You'll see another one popping up very soon, if I'm not mistaken, that's going to come, fly by, and there's nothing you can do. The breath hold is a key to the success of targeted deposition. So that was referring back to challenge 1. Challenge 2, and you've seen that already in the previous slide, is that if you think of a commercial nebulizer, for instance, since here our technology was implemented with aqueous suspensions, although technically you could have this concept of spion-loaded drugs could be implemented with dry powders. But the point I want to draw is if you look between the left distribution and the right distribution, is that commercial nebulizers fall in this 1 to 10 micron range, if you think of kind of the standard drug delivery approach, and it's important to minimize any superfluous deposition, which again we want to minimize, that you fall under this 1 micron range to avoid sedimentation and of course not be too small for high diffusion. That was regarding challenge 2. Okay, and what about challenge 3, the sedimentation problem? So we just wanted to show you that actually sedimentation for this particle size range, again, the half micron population, is extremely weak. So have a look. This is the bolus. We're talking. I mean, I'm talking with you. This is happening in real time. And it looks like it's a levitated system. I mean, it practically has kind of the feel of, I guess, a Star Wars laser beam that would be in suspension. And I'm going to go forward. These are about 30 seconds already. I'm going to go hop to 1 minute. Okay, this is after 1 minute. Okay, it's still completely levitated. Practically no deposition. 2 minutes. Here we are. It's still there. Okay, and I'm going to hop now just so you see. Practically at 4 minutes, the bolus is still there. It's not deposited. So these are way beyond the time scales of breathing, of breath hold that you possibly would imagine. So we're completely safe in terms of holding your breath and pushing out, and there's no chance for sedimentation during these time scales. We need is now, hang on, next slide, excuse me. So what you're seeing here is that we took true scale models of the trachea in gray, and for visualization purposes, we used 2 main bronchi of the right sizes, and you're seeing them with the lasers that are going to be shooting through them so we can see. One bronchus is going to experience the magnet, whereas the other one is not going to. And it's going to inhale and then exhale again. So here we go. And in real time. So all these are shown at actual speed. So you see the bolus is split. We purposefully put it against the side of the bolus so that the magnet would pull the furthest away, so you'd see it in action, and then the valves go back and you inhale. So once again, split of the bolus into the 2 main bronchi, deposition, and it's going to be exhaled. There you go. So what we did is the same quantification approach, which is have the transparent slides, roll them up into these transparent tubes, collect the fluorescent particles in both bronchi, and measure or visualize the deposition field. And what we saw, and which is stunning, is that one bronchus, the bottom one to your eyes, has not a single particle deposited, whereas the top bronchus has this patch that you see, again, using the same color code of this near its neighbors. In other words, you've basically nailed a localized deposition without having superfluous deposition in the sister bronchus. So this is extremely encouraging. All right. Now, we took the difficulty level to one more step up. And what we did is we 3D printed about 5 by 3, I'm counting 1, 2, about 3 or 4 bifurcations here that you see. These are 2 halves of a model, the top half and the bottom half. And the idea was whether or not by focusing that magnet to the specific spot that I've shown in red, you could get deposition there. So you're trying to aim for a localized deposition in this specific mid-airway, so to speak, without having deposition in the other regions. So we did this multiple times. So we ran for the visualization process, targeting, shooting about 50 boli so that we could collect something visual. And what you're seeing here in this inset is actually the deposition after about 50 target hits of the spions collecting in that region, in the region we were targeting for. Okay? So your eyes are actually visualizing where the bolus hit. So we quantified this, okay? And what you're seeing here in the color code is basically a ratio of the local deposition. We cut basically the airway into many, many segments. We counted under microscopy for each of these segments, airway segments, the number of particles. We looked at the particles per unit area, okay? And then we rendered a map of this distribution, so to speak, concentration of particles. And what we found in an encouraging way, I mean, your eyes were basically seeing this with the black stain of spions, is that you get something at the very least that's actually a 10-fold increase in local targeted deposition compared to the other regions. And that's very, very encouraging, because if you think kind of the best that was before was a 2-fold increase, here you have 1 order of magnitude at the very least in terms of improved localized deposition. Please bear in mind, and this is important because when one speaks in general about drug delivery and enhancing drug delivery, it should be noted that usually what you're aiming for is a relative increase. It's not perfect targeting. Targeted delivery by definition is a relative improvement against a gold standard. So a 10-fold increase is potentially quite dramatic. All right. A brief outlook, because here we just wanted to give you some pointers of the engineering technique. So in some sense, we're quite confident, I think we're very confident that the engineering solution holds the keys to get targeting correctly. Our in vitro experiments support this very strongly. And for us now, we're in the midst of working on ex vivo lung experiments so that our proof of concept is more realistic, of course. And the other thing that needs to be said is, what about the kind of clinically driven magnetic system? Of course, what you've seen in the in vitro experiments is bound to magnets that were touching the airway. I mean, we all agree here that we are striving to have so to speak, a non-invasive external activation mode. So the magnet is completely outside the body. So you need a magnet that's strong enough to be and focused enough with its design so that it doesn't need to touch the airway. It's kind of at a distance. So what you're seeing at the bottom right is this is what we had designed actually. It's a magnet that looks kind of like these rings that are in a pyramid shape of these permanent magnets, so that the magnetic field kind of looks like a shooting spike that's going to orientate itself in a straight line. And now we're working on these ex vivo experiments of these excised lungs with this external magnet to further this proof of concept before we hope one day to go into preclinical in vivo experiments. So with that, I'd like to thank you for your attention, and if there are any questions, I'll be happy to take them.
Thank you very much, Josué, for a fantastic presentation. I'd now like to open the floor for discussion. Remember to send us your questions using the chat feature on the bottom of the platform. Lovely. So the first question that we have is, how can the depth of targeting of the aerosol pulse inside the lungs be a priori known for a given patient?
So this is one of the recurring questions that we've often been asked. How do you see this in a clinically implemented way? Because how do you know a priori, and by that I mean, how do you know ahead of time how deep you should bring the bolus? So if you think of the cancer patient, and I mean here the lung cancer, most of these patients have undergone some form of a CT to identify the presence, of course, and the location of the tumor. And so with that, it means that the patient has a history file of the CT, which we can use as a benchmark to know the depth of the depth and the location of this tumor, so that we can calibrate what the machine, the smart inhaler, should use as a baseline volume to reach the target depth. That's the essence of it.
Thank you very much, Josué. If anyone does have any more questions, please feel free to use the chat feature and let us know. We have a second question here, which is, following in vitro experiments, what are the immediate next steps in furthering the technique?
So my outlook was kind of drawing the attention to this. I think, we are confident that the engineering recipe is there. A lot of the work now is almost at the level of the calibration, of the tweaking of what this ideal or optimized magnet will be. And so this is the step that we're undergoing with ex vivo excised lungs, is how to do the better combo of what right magnet with kind of a human-sized lungs. So it's, in some sense, it's got the twist of kind of an R&D approach at this stage, which is tweaking the engineering solution, because that part's already established. Thank you.
Wonderful. I believe that's all the questions that we have today, which brings the Q&A to a close. I want to say thank you very much for your questions, and once again, Josué, for this excellent presentation. This webinar is part of the run-up to our 4th annual Inhalation and Respiratory Drug Delivery Congress, which will be taking place on the 29th to the 30th of April this year in London, UK, as part of our Formulation UK series. If you would like further information on the Congress, or would like a copy of the conference programme, please contact the marketing department or visit the conference website. On behalf of Oxford Global, I would like to thank everybody for joining us today. I would like to say a big thank you once again to Josué, who has put a lot of time into making sure that this webinar was as interesting and valuable as possible. All that remains to be said is have a great day, and we look forward to welcoming you to one of our future events.